Yes. Yes. Aude.
So my lab is working on the evolution of immunity across domains of life. So all cells are infected by viruses. And when you think about it, these are phenomena that affect very different organisms, but in the end, there is always an arms race that drives the diversification of antiviral systems. And so when you think about this, one of the consequences is that when we think about different organisms, and by this I mean bacteria, plants, or humans, will have different types of immune system. This was pretty much what was expected and known for decades, that because of these ongoing arms race, you have different organisms, different antiviral systems. But this picture started to pretty much be proven wrong thanks to the recent unveiling of highly diverse anti-fatty systems. So in the past 5 years, more than 150 novel anti-fatty systems were uncovered, with really an incredible diversity of molecular mechanisms. And among the diversity of these molecular mechanisms, one of the striking observations was that some of these immune systems that exist in bacteria actually looked very similar to known immune systems in eukaryotes. And pretty much it was uncovered that there is an overlooked aspect of the evolution of immunity, and it's a conservation. And that you have genes such as the viperin or tear domains like tear domains, that pretty much share striking similarities with eukaryotic immune genes. And so somehow these genes have this paradox of being genes that needs to be able to probably evolve fast to adapt to new pathogens, while at the same being conserved across billions of years. And initially it started as an observation of a couple genes or a couple things, but this list has been growing rapidly, and today we are aware of more than 10 examples of conserved either domains or genes across of immune genes across domains of life. And so that's what my lab is focused on. So we started naming these ancestral immune modules, but overall what we explore is a conservation of antiviral immunity across domains of life, and what can we get inspired from eukaryotes to make new discovery in bacteria, in prokaryotes, or what do we know about prokaryotes that can help us discover new things in eukaryotes. And I will just tell you a piece of one story today. I will not kind of describe everything that we do, but you can ask me questions later if you want. I just wanted to share the story that is on bioRxiv, but will soon come out, about viperin immunity and how it evolved across the tree of life. So one key aspect, and one for me very question that bugged me since the discovery of the fact that these immune genes are conserved across domains of life, is how do they evolve? How do they remain relevant for immune mechanisms whilst having a really striking conservation? And so one of the key issues with this is that when you look at some genes like the sting, you actually see that it changed so much that it's very hard to actually do evolutionary studies. And a lot of these conserved genes, like all the tear domains, evolved a lot. So it's a bit of a hard problem to start with. But there is one exception to this rule, and that is of the viperin, which shows a really high conservation in sequence and structure. And so we decided to focus on viperin and see how they evolved to pretty much be able to understand a bit more about this topic. And viperin were initially discovered in human, and they produce a small molecule that is an RNA chain terminator, and that pretty much leads to premature RNA termination and antiviral activity. We discovered a few years ago that actually bacteria also have viperins, and that these viperins work in a similar manner. But we only had data points in kind of humans and mice and bacteria, but the rest of pretty much eukaryotes and archaea was a bit underexplored. And so what we asked was, where are viperins on the tree of life? How can we detect them, and how can we understand them? And so we did, I would say, a detection of all these viperins across domains of life, and found that they're really present in all domains of life, so bacteria, archaea, and eukaryotes, and they're actually much more common in eukaryotes than they are in bacteria. they are lost in specific clades like plants, land plants. But given this distribution that it's present in all these different organisms across the tree of life, it kind of suggested that they might have been present in really ancient organisms and have been protecting organisms for billions of years. We followed this with a phylogenetic tree of all these viperines, and when we put these three together, what we could see is that there are really the three domains of life recapitulated within one single protein, mostly with bacteria, archaea, and eukaryotes, with eukaryotes emerging from the archaeal clade. Interestingly, there were additional events of horizontal transfer of eukaryotic viperines from other groups of bacteria and archaea, but really one major event of acquisition here. And so one question could be that maybe these genes actually stopped being involved in immunity. Maybe they were involved in another function. So could we ask the question of whether the viperines, all these 1,300 genes, had a similar function across the tree of life? And this is a hard question because actually they are present in really a lot of diverse organisms, these viperines, and a lot of organisms we cannot grow in the lab. So how to tackle this question at scale? Well, here is the trick, is that viperines are so conserved that if you are looking here at what we call a plaque assay, where we have bacteria and we put phages, so viruses that kill the bacteria and are able to infect here, if you express the human viperine, so human gene in the bacteria, it's able to protect, to function, to produce a small nucleotide, and to protect the bacteria against phages. And so we decided to take advantage of this because we can also go a step further with this method and do a biochemical characterization to actually try to express a viperine from really diverse organisms across the tree of life to understand whether or not they would have a similar function. And we could actually see this type of antiviral activity in super diverse organisms, some of which are represented here. We can even go further and actually characterize the type of molecules produced by these viperines through this setup. And we could show that actually while Asgard viperine and archaeal viperine are what we call promiscuous, meaning they produce different type of molecules in a similar way as bacteria do, the eukaryotic viperine are substrate specific. They produce one or the other molecules. And so we wanted to go a bit further and to try to understand whether this specificity emerged and was selected for. So could we explain the evolution of the substrate selectivity? And to do this, we actually folded viperines. Viperine is the first protein I folded with AlphaFold, like a prokaryotic viperine. And we actually folded a bunch to try to follow this. And we're able, using this prediction of different structures and also structural guided alignment, to find three different types of pockets: promiscuous pockets, CTP selective pocket, and UTP selective pocket. And this was just observation from analyzing predicted structures. And interestingly, they mapped our experimental results, meaning that when we asked which of these pockets would these viperines have, we found that they would have the ones we had predicted to be CTP selective or UTP selective. And so then, armed with this conviction that this was working out pretty okay, what we did was to actually do a structural guided alignment and map the type of pocket we would observe on the viperine tree. And really, in a kind of cool manner, all of the promiscuous pockets are here, and then we have CTP selective pocket and UTP selective pocket. And we can really map the emergence of substrate selectivity here after pretty much in the major clades of eukaryotes. Interestingly, there is one eukaryotic viperine which has a promiscuous pocket, and that is here. And when we looked at it, we estimated that actually, yes, it is experimentally promiscuous. And then what we did was to pretty much try to reverse it and to make it CTP selective. And when we did and modeled this, we hypothesized that we can do 2 amino acid changes and change the pocket from promiscuous to CTP selective. So actually, we introduced this mutation and measured experimentally what was going on in terms of production of molecules. And we could indeed show that these 2 mutations restored somehow a CTP selectivity, showing that we are just kind of 2 mutations away between promiscuous and CTP selective in this. And the fact that we only see CTP selective in this context really strongly suggests that there is a selection for this CTP selectivity. And so overall, using these viperins, we were able to shed light on the evolution of immunity in eukaryotes. And I think it's one really exciting way of studying immunity in things like early eukaryotes, but also in non-model organisms where immunity is really hard to work on because we don't have the model that we can grow in the lab and infect, etc., etc. Here, we're really breaching this and using this type of methods, using bacteria as a model to study immunity across domains of life. And of course, we're now wondering whether other shared immune genes evolved in a similar manner, what drives the retention of specific immune systems over other, and other cool things I did not have time to discuss about genetic linkage in eukaryotic immune genes. And on this is my lab, and thank you for listening.